Assalamualaikum and very good day to an honorable lecturer, Dr. Hadijah binti Ibrahim and classmates. We from Group 1 consists of Mat Nazim Razali bin Kanini, Muhammad Aliff Amzar bin Othman and Muhammad Afiq bin Shah will present a case study from Chapter 8, Collaboration Strategies, Ending HIV, Sangamo Therapeutics and Gene Editing. Sangamo Therapeutics is a biotechnology company based in Brisbane, California. It uses cell and gene therapy to treat hemophilia and other genetic diseases. The company was founded in Richmond, California in 1995. It was formerly known as Sangamo Biosciences Incorporated before changing its name in 2017. It employed 182 people as of September 2018. Sandy McRae is the president. In 2018, Edward Riba was appointed as Sangamo Senior Vice President and Chief Technology Officer. Sangamo focuses on translating science into genomic medicines that improve patients' lives by utilizing platform technologies in gene therapy, cell therapy, genome editing, and genome regulation. What are zinc finger nucleases, ZFN? It is a technology that could edit the genetic code of a living individual to correct genetically based diseases like hemophilia, sickle cell anemia, Huntington disease, and others, or confer genetic resistance to non-genetically based diseases. ZFNs work by snipping DNA in a specific places. The cell then repairs the cut by polishing the two ends of DNA together or copying the corresponding sections of DNA in the other chromosome pair. These homologous substitutions from the other chromosome corrects the faulty gene. A single gene defect causes monogenic diseases. One is hemophilia. Hemophilia, lack of clotting factors, causes more bleeding after an injury. Sangamo had shown that its ZFN hemophilia treatment method worked in mice and was preparing to apply for clinical trials. Sangamo treated sickle cell anemia, beta thalassemia, and other monogenic diseases. Sangamo treatment could kill the BCL11A gene that causes these diseases in the lab. Huntington disease, HD, is another example of a monogene. The disease impairs motor coordination, cognition, and memory. The disease is caused by a single copy of the faulty gene in most cases. In addition, 50% of HD patient children are affected by the disease. Despite previous research into ways to reduce the protein cellular presence, it was discovered that mice lacking the normal Huntington protein die before birth. This method enables Sangamo to isolate and turn off only the faulty gene. A person can produce Huntington protein even if they only have one functional copy of the gene. As a result, Sangamo announcements of promising results for its HD treatment was huge news. Drug development is both expensive and risky. According to most studies, it costs at least $1.5 billion and a decade of research to bring a new Food and Drug Administration's FDA-approved pharmaceutical product to market. Accounting for investment in failed drug efforts suggests that drug development costs are much higher than is commonly reported. A study of R&D spending and new drug approvals published in Forbes in 2012, for example, discovered that firms spend more than $6 billion per approved drug. Sangamo also threatened that other gene editing technologies would render its ZFN technology obsolete. Talents also known as transcription activator like effector nucleases and CRISPR-Cas9, also known as clustered regularly interspaced short palindromic repeats, gained popularity in early 2018. Talents, like ZFNs, are nucleases that recognize and bind to specific parts of DNA before cutting the genome at those locations. The difference is where the DNA binding site is located. On the other hand, talent technology was easier to design treatments with, giving it a longer-term advantage. CRISPR technology used bacteria natural defense system to detect and eliminate foreign DNA, providing bacteria with adaptive immunity. CRISPR was even simpler and more efficient than talents, sparking great interest in its potential. CRISPR-Cas9 was so simple and inexpensive that high school and college students were taught gene editing in the classroom. However, because CRISPR relied on a short RNA sequence to guide its activity, some were concerned that it would result in unwanted off-target cleavage. These two threats pose both benefit and cost to developing ZFN technology. The theme of the chapter, collaboration. Biotechnology companies like Sangamo rely on research grants and collaboration agreements, and almost all revenue is used for R&D purposes. The result of this study can provide the latest and radical treatments for patients' recovery. However, without financial support, resources, skilled workforce, and market capability, the results of this study will not be marketable and usable. Sangamo only had less than 200 employees in 2019, limiting their ability to conduct the clinical trial, production, and marketing. For example, based on sizzlingstock.com, Sangamo's market cap for 2006 was $0.23 billion USD compared to Johnson & Johnson, the world leading pharmaceutical and biotechnology, with $179 billion USD market cap for the same year. So, in developing their innovation and technology potential, Sangamo took the approach of collaborating with several firms. Since its inception, Sangamo has collaborated with Gilead and Case Western University. The collaboration with Gilead is for using ZFN for cancer therapy with a total of $150 million, while CW University is for research grants mainly related to genome technology. Sangamo also has an exclusive global collaborations and license agreement with Pfizer Incorporated for gene therapy programs for hemophilia A, ALS, and FTLD, with BioVirative Incorporated for hemoglobinopathies including beta thalassemia and sickle cell disease, and with Shah International GmbH to develop therapeutics for Huntington disease. In addition, Sangamo has established strategic partnerships with companies in non-therapeutics applications of its technologies, including Sigma Outreach Corporation and Dow AgroSciences. With this collaboration, Sangamo has increased its market cap to $0.98 billion for 2021, an increase of almost 5 times compared to the initial year of its establishment. The study and use of ZFN technology entered a new phase, with Sangamo beginning conducting R&D related to HIV treatment in 2018. The idea was to remove a patient's CD4 T-cell, modify them in a lab using a zinc finger nucleus to render the cell CCR5 gene defective, and then reinfuse the modified cell back into the patient. Handicapped CCR5 will not permit HIV to replicate. Collaboration to enable genetic therapy for the treatment of HIV has been done, including the success of obtaining a research grant of $11 million from Case Western University. Now, Sangamo is actively looking for partners and collaborators who can make a successful gene therapy program for HIV. Now, I welcome my team members to present on the latest developments from Sangamo Therapeutics and discuss questions from the case study.